Hey everyone, welcome back. So in our last few videos, we have been using pure Python to solve real-world lab problems like calculating melting temperatures for PCR. So today we are going to solve a massive biological mystery using a little bit of math and a Python sliding window. So bacterial genomes are massive circular rings of DNA, right? So when a bacteria wants to divide it, it has to copy that entire ring. But where does the copying machine starts? So out of millions of identical looking letters, uh, how does the cell find the start button, also known as the origin of replication, right? Or ORIC for short. Okay, so it turns out because of the physical way DNA is copied, one side of the circle becomes rich in guanine, which is G, and the other side becomes rich in cytosine, which is C. The exact spot where this flips is the original. So to find it, we calculate something called the GC skew. Okay, so let's jump into VS Code and write the algorithm to find it. Okay, so the maths for GC skew is incredibly elegant. So in any given chunk of DNA, you count the G's and you count the C's, then you use this formula. So GC skew is basically equal to G minus C, then divided by G plus C. Okay, this is the formula. Let's move it into Markdown. So if there are more G's, the number is positive. If there are more C's, the number is negative, according to this formula. Okay, so the exact point where the graph hits its absolute minimum is our start button. So let's write a Python function to calculate this. Okay, first let's create a dummy DNA sequence. I'll engineer this string so it has a high concentration of C's in the first half and high concentration of G's in the second half, which will mimic a real bacterial genome. Okay, so first of all, we will import our library. We are going to use matplotlib dot uh, pyplot as plt. Okay. Then what we will do is we will take a pre-made basic 100 page dummy sequence with a hidden flip in the middle. Okay. Then we can simply print it. We can print the genome length of DNA genome, right? And we can run this. Now, we don't want to calculate the skew for the whole genome at once, obviously. That would just give us one number, right? So we want to use a sliding window, just like we did in our Kamer video, okay, to see how the skew changes over time. Okay, some mistake happened here. Yeah, length of DNA genome, this will be a circular bracket, I believe. Yep, this has to be a circle bracket. And that should work, and there you have it, right? So but we don't want the single number, so we are going to use a sliding window, okay, to see how the skew changes over time. So for that, what we are going to do is we are going to define a function which we can call as calculate gc skew. Okay, and the parameters that we will pass will be the sequence and the window size. Okay. Then what we will do is we will calculate GC screw across a sequence using a sliding window. So basically what we are going to do is we are going to call this Q values is equal to, and we are going to leave this blank. Okay, not Q value, we will call it Q values. Right. After that, we are going to run a simple for loop. So for I in range length of sequence minus window size plus 1. Okay, and inside that loop we will use window is equal to sequence I to I plus window size. Okay, then we will calculate G count and C count. G count is equal to window dot count and we will count G. Similarly, we can copy this code and we will call it C count, and instead of C, G will count C. Okay, that should do it. Now what we will do is we will write a code that prevents division by 0 if a window has no G's or C's. Right, that is an edge case that can happen. So what we will do is if G count plus C count is equal to 0, at that moment what we will do is skew values dot append 0. That should do it. And we have to add an else condition as well. So else what we will do is we will apply the GC skew formula. So skew is equal to G count minus C count divided by G count plus C count. Okay, then finally skew values dot append skew. Okay, and we'll come out of the loop and we will finally return the skew values. Okay, that should do it. So now let's try to run it with a window size of so let's say 10. Okay, so we are going to call it skew data is equal to calculate GC skew, and we are going to pass our DNA genome, right? DNA genome, and our window size we are going to use 10. Okay, and finally we can print calculated length of skew data is skew data points, right? And let's run this, and 67 comes. Okay, so we have calculated 67 skew data points. Now we have the data, right? But finding the absolute minimum point in a massive list of decimals is a job for Matplotlib, which we have already imported. So let's plot our skew data array as a line graph, right? So plt dot figure. Let's use the figure size. Let's go with 10 and 5. Okay, now we will plot the skew value. So plt dot plot skew data, then color. Let's use what should we use? Purple. I think the P might be small over here. We'll check. Then line width. If I'm not wrong, that should be the command, and we'll use 2. Okay, now we will add a horizontal line at y is equal to 0 for the reference. So what we will do is plt dot what's the command? Axh line. Yes, axh line. Then 0. Let's use the color black, right? And we will also use line style as Let's use a double dash, and alpha will be 0.5. That should do it. We will check it after running it whether it runs correctly or not. Then finally we will have our min skew, which will be minimum of skew data, right? Then we will have a min index, which will be skew data dot index of minimum skew. Now finally we will draw a red dot should be fine. Exactly where the start button is. Okay, so plt dot scatter min index, then min skew, color we will use red, then s is equal to 100, then the z order is equal to 5, then label we can say, what should we say, origin of replication, right? And finally, time for some label to make the plot look attractive. Plt dot title, we will call it this is skew across the genome. Let's use font size is equal to 14. Font weight will be bold. What next? We'll have to use X label. We'll call the X label as genome position. Let's use font size as 12. After that, we are going to use plt dot y label. Y we will label as gc skew. We can also mention the formula over here just in case, right? That should do it. Then for font size, we will use 12 again. Then finally, we will plot the legend. Right, that should do it. Let's just make it a tight layout. All right, and finally plt dot show. And there you have it. Look at that beautiful chart. The purple line starts off highly negative because our sequence was flooded with cytosine, right? But right in the middle, it abruptly shoots upwards into positive territory as the guanines take over, right? That giant red dot at the very bottom of the V shape, that is the absolute minimum GC-skew. If this were a real E. coli genome, you could tell a lab technician, look exactly at position something, right? Position 38 or something, right? That is the physical location of the oriC gene where DNA copying begins. And there you have it. By combining a simple Python sliding window with Matplotlib, you just built a sequence analysis tool that solves a fundamental problem in microbiology. Algorithms like this are the backbone of computer biology. And you don't always need massive machine learning models. Sometimes you just need a brilliant mathematical formula and a simple for loop. The code will be available on GitHub as always. Try changing the window size to 5 or 20 and see how it smooths out the graph, right? So please hit the like and subscribe button, and I'll see you in the next video.